So I would first like to thank everyone for attending this MDPI International Day of Plant Health of 2025 webinar. I am Zorana, and I'm actually a journal relations specialist for the journals that are supporting this webinar, and I'm here to moderate this webinar. So any questions that you might have, any technical problems, you can always contact me. Here today we have wonderful speakers and prominent scientists who will speak on their topics, and I will just give you now a brief introduction on what to expect during the webinar. So first of all, I think you're all very familiar with MDPI. We have been around since 1996, and we are based in Basel, Switzerland. We now have over 470 diverse open access journals, and we are actually trying to be very open with our mission to share the knowledge openly and to make it accessible to everybody. So also, we would like to present something that is within the topic of this webinar, and this is the third International Online Conference on Agriculture that we're expecting to have in October. You can have more the information about this conference on the webinar website as well. But here are some of the highlights that you can see. The conference chair is Professor Dr. Bin Gao, and you can see some of the important dates. For example, abstract submission in 24th of June and registration for the conference in 15th of October. And these are some of the sessions and topics that are covered during the conference. Please know that this is something that could be very useful for you. Usually the registration is free and all of the information can be found online. When it comes to our program today, we will start with the presentation of Professor Dr. Horacio Heisen, and it will last for 25 minutes. Then we will go to Dr. Azucena Gonzalez Colomna, then to Professor Assunta Bertaccini, Professor Dr. Hikmet Budak. And after all of these speeches that will last approximately for 25 minutes, you as antideans can join us on the Q&A session where you can ask your questions to the speaker about their presentation. Please, between the Q&A box and also the chat box, it's actually very easily said in, I will just show you, it's very easily said a QA box. This is where you will write your question instead of a chat. It's important to know because we will be able to see, and speakers will be able to see only in a QA session. And also, please be patient. We will take turns with any questions, and make sure that you write the name of the speaker who you would like to ask the question to. Before we move to the speeches, I would also like to tell you that we have some relevant special issues from multiple journals also selected, and you can see some of them here. But you can also find the information about this special issue, deadlines, and guest editors at the conference and the webinar website online. I would not like to keep you more waiting. So I would just like to go to our first speaker, which is Professor Dr. Horacio Heisen. He's a full professor of pharmacognosy and natural production at the Faculty de Química University in Republic of Uruguay. And his research focuses on analytical methods for food, environmental safety, sustainability, and natural product analysis using NMR and mass spectrometry. He has led more than 25 international training courses and has held leadership roles in LAPRW, the Sociedad Latinoamérica de Fitoeconómica, EUPAC, and more. And I would now like to please welcome Professor Heisen with his presentation.
Okay, thank you for MDPI for the invitation to give this to participate in this interesting webinar. I will first introduce our group. We are a group in Uruguay, in South America, the tiny country between two giants, and we are a multi-center laboratory that works coordinatively in a coordinate way, because our instrumentation is very rather poor, so we have to share it and to we analyze a lot of different matrices for pesticide residues, contaminants, and also there is another line of work that is on natural products, but today I will focus on our work on the contaminant parts. That is the GACT, is the acronym for Grupo de Análisis de Contaminantes Traza, o sea, the group for the analysis of trace compounds. And today I will try to introduce you. As a first speaker, I thought I had to talk something about One Health and plant health, and then how to protect plant health and the consequences, sometimes protection products to ensure the plant health for the One Health paradigms. Something about trace analysis. I will show you some results, not entering too many details on how these analyses are done, some on a simple overview. So we are in the International Day of Plant Health, and the theme day is the importance of plant health in One Health, as you already know. And the inclusion of plant health within the One Health paradigms is a joint proposal of FIO and the International Plant Protection Convention that has the objectives of a sustainable agriculture to protect the environment and facilitate trade and fair trade through the promotion of harmonized scientifically based phytosanitary measures, and mostly developing phytosanitary capacity for members to accomplish these three objectives. So the rational use of the plant protection products mainly, and the use of the agroecology concept, for instance, or IPM concept. So the idea is to protect farmers from economically devastating pests, to protect the environment, protect ecosystems, industries, and facilitate trade. And as a result, it will protect livelihoods and food security by preventing the entry of new pests into a country, because nowadays, as the FAO says, pests fly with the planes. So the borders are very, very diffuse nowadays for one or other pest. So One Health, in summary, is a collaborative, multisectoral, and transdisciplinary approach that recognizes the interconnectedness between the health of people, animals, plants, and the environment. And the key word here is interdisciplinary, because in any of the task itself is a very, very broad one, and only can be accomplished if everyone collaborates among with other. So the One Health concept has evolved since it was a three compartment: healthy people, healthy animals, and healthy environment by itself. But I think it's if you think in a football ball, then the grass where these three concepts run is precisely plant health. So, because as it says in the FIO videos, abundance brings prosperity, but there are threats that diseases or pests can be very dangerous for crops and for food. So that for that reason, the plant system is crucial for accomplishing the One Health concept. So let's see, for instance, you see here a very nice river, but if you water important component of of the environment and the produce, because if you see here, when you use reclaimed water for irrigation in greenhouses, then you can see that the produce can get some emerging contaminants, some pharmacs that are also found within the tomatoes or the cucumbers and peppers you analyze. So it's a worldwide concern. The good quality of the things you use for producing our food. And plant health and one health are, of course, intimately, as I said, connected, because that is taken from the FIO website, Plants Are Life, and 80% of the food we eat and 89 of the oxygen we breathe come from plants. But the loss to pests and diseases is very hard to sustain, and we have to optimize this production. So the key is zero hunger, and that is food security. And for food safety, it's a crucial part of the food security. Last year, the motto for the World Food Safety Day was "Prepare for the Unexpected," because we are in a global world, and there are no borders for the disease or the chemicals that can spread from one to the other. So, and the difference between an agricultural product and a food is their safety. That is a crucial concept that I will develop in the coming slide. So, why plant health is relevant to One Health? Okay, plants give food for us and feed animals, but when to protect these crops, we use plant protection products or pesticides that are a threat to the environment also, because if they are not properly used, because once the pesticide is applied, it reaches the whole environment, and there are perhaps there are the target organism in the crop, but also beneficial insect, terrestrial organism, water organism. It can go into the drinking water, so it goes from the emission to the distribution and then the exposure of the fauna, flora, and also the human beings. So we have to deal with the pesticide toxicity, because they are molecules that are designed to kill, and we have to know their toxicity, and we have to be aware also that the effect that they could have, it's not it's measurable in a very broad time range. So we have to be very aware of that. That was dramatically seen in the butterfly effect that we saw with the DDT, that it seems, if you look at the molecule, at the DDT molecule, you see it's like an insect, but it was the residues of DDT, due to improper use, put in danger all the fauna and diversity that led to their banning in all over the world. And the issue is the pesticide residues that are defined by the World Health Organization as any substance or mixture of substance in food or for animals that come from the use of a pesticide. And we have to be aware that it is important, not only the parent compound, but also everything that can be considered of toxicological significance. The key, one of the older examples is the change from malathion to naloxone in the early 80s, that it the metabolite is even more toxic than the parent compound. So we have to know how do pesticide residue behave in the environment, and that is a complex puzzle where you have to develop analytical method and understand plant metabolism, animal metabolism, how these these products will affect plant growth and development, to know about phytopathology, perform some feeding studies, field trials, to see how it works in fields, and then after that, the processing. So we have to have a clear idea of the pesticide residues occurrence in the environment, food, and feed, going back into our One Health concept. But what does a residue mean? Imagine a container and a fly in the container. And a fly in the container means 3 ppm. We have to analyze and detect MRLs where the quantity to be determined is about 0.01 milligram per kilo. So to ensure plant health within the One Health concept, the use MRLs is a good starting point, because MRLs are based on good agricultural practices that you have to say when to apply, how much to apply, and how many times. That is the key for a toxicological safe agricultural product that can become a succeeding food. And the MRLs are defined after many animal studies and corrected for safety factors, and define the acceptable day intake and the acute reference dose. With those parameters, the MRLs are set, and we have, as an idea, when MRLs is originally defined as a trade concept, but it's also based on toxicological findings, and the MRLs is very low compared to the LD50 because of the safety factors I already told you. We have a wide world of possible contaminants, veterinary drugs, pesticides, and to see, for instance, in the case of analytical, the analytical target we should have, that is around 150 and 500 pesticides, and we have to look for these compounds. And how to do it? We have to perform what we call a trace analysis. That is the analysis of minor components in an homogeneous mixture. And as the sensitivity of the instrumentation goes lower and lower to lower levels, then the concept has evolved from nanogram to gram or nanogram per kilogram and ultra trace level. So that's our task. That is moving the border or the horizon is moving continuously. And we have to move within the modern analytical concept that use less reactives to produce less chemical waste, and that is supported by enhancement of the instrumentation sensibility that leads to the miniaturization. And how to find it? Then you can fish with net, or you have to fish with reels. One is multi-residue method. The goal is to determine as many residues as possible in one shot or two. And the mono residue methods are one residue each time. As if we consider the amount of chemicals we have to analyze, then we have a lot of, we cannot do it one by one, but there are some compounds that need a specific so to say, methodology to determine. And the analytical methods are developed and validated according to the need are fit for purpose. And we need to yield confident, reliable, and reproducible data on the presence of pesticide residues in the samples at the required level, that is mostly the MRLs. And for that, we have instrumentation, a world of molecules. We need knowledge, quality assurance, and quality control. The results should be representative and that be supported by the regulation. So we need accurate and precise, reliable and robust, and based on the guidelines, the analytical guidelines that are accepted by the regulations. The concept, as I said, is to get methodologies that are fit for purpose. And we have to look after the validation parameters once we find a possible method to determine the trueness, the precision, the LOD and LOQ, the linearity, and the matrix effect. With this method in hand that covers all these aspects, we can face now the analysis of our task. So let's go now to plant health and environment. For instance, you have to look for the herbicides in soils. Herbicides are the most employed plant protection products in the world at this time. And here is how we try to analyze using ionic chromatography to determine glyphosate, AMPA, and dicamba that are one of the herbicides that are applied together with glyphosate. Or also how the use of pesticides impact in bees. That is a major problem worldwide. And here I show you one bee sample that has 8 pesticides in it, and we can see that the content of pesticides in the hive is depending on the time of the year we collect the sample, and that is during harvest or post-harvest or pre-harvest, the amount of pesticides are dramatically different. So we develop a method and validate it. We use almost the Ketchers method, that is the most popular one, and we look for all the parameters I talked already, and we look for the recoveries. Here is, for instance, at 3 different levels for around 50 pesticides in this case, what I am showing you, and then we look everything is okay, and we use it, for instance, to see how the pesticides applied behave on the field at the label doses. For instance, in the case of citrus, to prevent citrus diseases that are one of the major threats for citrus cropping. So we tested, for instance, in our country, 2 different regions to see if the dose level is enough to accomplish the MRL for one, and how it decays during time in order to have a good protection of the product. But we also look some seasonal monitoring. We have already done a lot of samples since 3 years in this time, and here I present you some findings on the Cucurbitaceae family. That is chlorpyrifos. For instance, we found 2 pesticides at the beginning, chlorpyrifos. Now chlorpyrifos is banned in our country, oxaliotrin also. And we found through the years eight active principles. One is some of them are allowed, other not. There are some issues to be corrected, and that's one of the purposes of the monitoring. And we found in this case from Zapallito, that is a variety of Cucurbita maxima that is very consumed in our country. One residue of pesticides within the scope of the method was detected during this methodology. And then we can look how risky is the presence of this pesticides in the produce. So we have three categories: chlorpyrifos and prosimidon, that are whose levels are above the MRLs, deltamethrin, pyriproxyfen, and cyalotrim. We do not exactly how to do, because there are some pesticides, some regulator bodies like the Codex that has one MRLs, and also the EU, for instance, has a different MRL for the three, and the other three are below the MRL, so there is no possible risk. So we look at these ones and trying to see the differences in the MRLs. We look to the mode of exposure, and then we use the formula that is the usual one, that is the no observed adverse effect level divided the level of exposure and the sum of them, and there is borders that are fixed by the regulating bodies, and in particularly chlorpyrifos and cyalotrin are complex molecules, so to say, and performing the study I showed you 2 slides ago, we see that there are some pesticides whose margin of exposure can be dangerous when we eat for these chemicals in these products. We also did it for onion, but in onion the situation was better, so to say, looking at the margin of exposure. Although there are some valuables that are for instance, here azoxystrobin is rather low, but is no genotoxicity potential, so it's okay, and it's below the MRL, so there is no problem with that. And then we go to the strawberry case, that because strawberry are one of the most contaminated fruits all over the world, up to 22 residues in one sample were detected in USA. And after 3 years monitoring Uruguay, we found the similar tendency that is reported all over the world. So we found a number of compounds per sample, and some of them showed MRL exceedances. So there is something we are working with the authorities in order to get a better result. But also we can have to look at some products that were the feed of the animals is important, because the final product is a very valuable food like milk. And then we look and try and develop methods that are multi-class, that can analyze not only pesticides, but also veterinary drugs and mycotoxins. And for instance, that was one case we did it with the contaminated feed because of early application. Chlorantraniliprol is a very a rather persistent agrochemical. Aflatoxin B1 could be present in the feed, but then if aflatoxins are present in the feed, then you can find aflatoxin M1 in milk, that is a metabolite of aflatoxin B1, and if some antiparasitic was employed, then it's possible to find ivermectin in milk, and that was our case in a seized sample of milk. So just arriving to the conclusions, let's see that I try to show you how plant health is constitutive of the One Health concept, and protecting plant health must not imply hamper the environment. Sustainability and good agricultural practices lead farmers for a proper use of plant protection chemicals. The dynamics of pesticides in the environment and the agricultural product should be known in advance before applying, and food safety is main concept for food security. Plant health and One Health are the basis to accomplish the SDGs. So that's what all I want to tell you. Just show you our team that is in Uruguay. I thank you for your attention, and I hope you visit our country, small but with some beauties that we are proud to share, and that is.
Thank you so much, Professor. I would just like to thank once more Professor Dr. Horacio Heisen from the University of Republic in Uruguay for sharing his presentation and knowledge on ensuring plant health within the One Health paradigm. I would like to thank you once more for accepting to be the speaker and for also giving this wonderful speech. We will now move on to our next speaker, who is Dr. Azuena Gonzalez Colomna from the Institute of Agricultural Sciences in Spain. And her topic will be on pesticides problems and innovative solutions in the context of the EU market. So Professor Colomna and her line of research focuses on the biotechnological production of the botanical and fungal biopesticides. She has published more than 200 scientific articles in index journals and also elaborated patents, and has been a mentor and directed over 14 doctoral theses among other contributions. So please, Dr. Colomna, when you are ready, you can share your presentation and we can start with your speech. Thank you so much again for joining us.
Okay, thank you. I think I'm sharing already. Can you see it?
Yes, we can see it. Everything is perfect.
Okay. Like you said, my talk is on pesticides, problems and innovative solution in the context of the European market. So, sorry. I work for the Spanish Research Council, which is the largest research organization in my country, in Spain, and we are located all over the country. Specifically, I work for the Agricultural Science Institute in Madrid. I am the leader of the biopesticides group at this organization because we work on natural pesticides from plants and fungi. Are really pesticides necessary? Yes, because they are necessary to protect cultivated plants from pests and diseases. Without pesticides, which are plant protection products, crop losses will range between 30 and 50%. However, as you have heard before, pesticides harm people, other organisms, and the environment because of their persistence and accumulations and everything that you've heard in the talk, in my previous talk. Nevertheless, the pesticide market is very large and is segmented by different kinds of active materials like fungicides. Fungicides are a very important market worldwide, and the percentage of growth until estimated for 2029 is growing at 5% during this forecast period. If you look at the insecticide market, the growth is even larger, and it's about 9% all over the countries. And Europe is growing at the same rate as the other countries. It's not there is not a big difference for Europe. or North America. Global nematicides market follow a similar trend with a lower increase, a 4% increase, and also Europe is at the same size as the North American or other regions in the whole world. The herbicide market is probably the largest segment of crop protection chemicals, is growing at 6%. And again, probably in the largest market is the US because of the genetically modified plants where they grow crops genetically modified, the herbicide inputs are larger. In Europe, the pesticides are regulated within a very strong regulatory framework, which is based on the Directive 2009, which is about pesticides, and the regulations about plant protection products on the market and the residues. These strong regulations resulted in the adoption of national action plans to reduce the risk and impact of pesticides, to promote low pesticides and agriculture by implementing the integrated pest management plans, and to ensure that the aquatic environment, drinking water supplies, and protected areas are safe from the effects of pesticides. So taking all this in account, we have the prohibition and restriction of the use of pesticides and active ingredients, which is a good outcome, but at the same time, it requires a desperate increase in new safe active ingredients. Therefore, there is a strong need for effective and safer alternative products. These products could be biopesticides from plants or microorganisms, which are the main sources of biopesticides. And in my talk, I'm going to talk about biochemical biopesticides based on natural products for controlling crop pests and diseases. Which are the advantages of biopesticides? Lower toxicity. They affect only the target pest and closely related organisms. They are less persistent in the environment, and their use in integrated management practice can reduce the conventional pesticides while maintaining the crop yield. However, in Europe, the strong regulation does not match the procedure for the approval of plant protection products. Why? Because biopesticides follow a registration process which is risk-based as any other active ingredient. And the biopesticides are regulated as plant protection products in Europe. So the active substance must be approved for the European Union before the plant protection product can be approved in the member states. So the procedure requires that approval of the pesticides, active substance, and then the approval of the plant protection product. So as you can see here, this procedure is very long. Like from the submission of the application and dossier to the first approval can take up to 10 years. So once the substance is approved, then the application for the authorization has to take place until its publication. So they require a dossier of the active substance with scientific data, toxicological data, and so on. They need a preliminary assessment report. Then the European Commission submits a review report, and then the subsequent authorization of a plant protection product is carried out. So this is really a long and complicated process, and also expensive. Sorry. So the EU basic substances and low-risk substances could be an alternative. Basic substances are not plant protection products, but they have the protection properties. The approval is based on existing assessments carried out in accordance with the legislation. Those low-risk substances have a series of regulations, and for a low-risk substance, it takes about 15 years for the first approval. For a normal substance, it takes 10 years, but then they need a renewal every 15 years in both cases. But for a basic substance, once it's submitted, the first approved, then it's unlimited, valid for an unlimited frame time. There are only a few basic substances and less low-risk active substances list, as you can see here. So that can give you an idea of how difficult this is to obtain in Europe. Regardless of all this regulatory framework, the biopesticides market in Europe is growing steadily. And among the European countries, Spain is one of the largest growing countries in the development of biopesticides. What is the context of our work? We work on botanical and fungal biopesticides, and the approach is that plants and their endophytes are chemical factories, and we are going to use those compounds for the protection of crops. For example, if we talk about active plant products, I'm going to highlight the ones we have been working on. From endemic plants, such as Persea indica, we have obtained ryanodines, which are very specific against Lepidopterus. Another endemic plant, like Betancourtia palmensis, we have obtained terpenes such as sylphinenes, which are very specific against beetles and very active and acting on GABA receptors, which are resistant to insecticides. From another endemic plant from the Iberian Peninsula, which is Lavandula luisieri, we have worked on essential oils which contain a unique type of compounds in this genus, Lavandula, which are necrodines and are very active on aphids, on acaroi, and also on plants, like weeds. Also, we have been working on active products from agroforestry residues. For example, residues from the production of essential oils, such as hydrolates. Hydrolates from the distillation of garlic, which are very active, which contains diallyl trisulfide, methylallyl trisulfide, are very active on nematodes. Also, the residues from the production of wormwood, we have obtained a residue with octadiendiol, very active on nematodes. Pruning residues from plants like juniperus, and also fruit trees, and also other trees. We have obtained a lot of different terpenes, which are very active on acaris and also on aphids, like from eucalyptus tree and so on. And we have developed a series of formulations. Also from bulbs, residual bulbs from the production of crocus, which is as saffron, we have obtained quincenoside molecules, which are very active against all kinds of Lepidopterus, beetles, and aphids. And also from the production of olive oil, which creates a lot of residues. These residues have been biotransformed, and we have obtained an extract which is active on nematodes and aphids. So you can see there's a lot of cheap materials to work. Also, we are worked on endophytic fungal active products. For example, from the plant Persea indica, we have obtained an endophyte Phyllosticta species, which fermentation gave from the deoxolanones, which are being characterized for the in vitro and in vivo activity against nematodes and aphids. Also, from the endemic plant, the Betancourtia palmensis, we have obtained an Aspergillus species, which fermentation gave aspergillic, neo-aspergillic, and melaine, which are antifungal and also active against ticks, very active. Also, from an Artemisia absinthium variety that we have developed, we obtained an Stemphyrium species, which fermentation gave stepholones and stemfol, very active on aphids. If we are obtaining plant products and fungal products to protect crops, we need the production of this biomass. For endemic plants, we have developed aeroponic cultivation, such as, for example, Persea indica. We obtained chemicals from the aerial and the roots, active on Lepidopterus, as we did with the wild plant. For endemic protected plants such as Bethencourtia hermosae or Artemisia granatensis, these plants are endemic and protected. Therefore, we need biotechnological production. We did the in vitro production of transformed roots, and we obtained African annuals from Bethencourtia and Artemisia granatensis. We obtained polycyclic acetylanes very active on aphids in both cases. We also have carried out domestication and field cultivation of selected plants, such as Artemisia absinthium, a selected chemotype. We have been domesticated for 12 years. We have registered a variety which is Candial, which is two-john-free, and we have developed antifungal extracts rich in linalool, epoxyocimene, and chrysanthemol. This is all patented. For the sustainability, also in looking into other endemic plants like Artemisia pedemontana, we did the aeroponic cultivation, and we obtained essential oils very active on insects and nematodes and ticks. We did the in vitro cultivation of Nepetateidea, endemic and pericalis species, and we obtained the Nepetateidona active on Lepidopterus, and all these benzofurans which were active on Lepidopterus from these endemic plants with sustainable practices. From domestication and plant and escalation in the field, we also have cultivated the medicinal plant Geranium macrorhizum for the production of germacron-rich oils active on ticks and aphids. We have produced Satureja montana, which is an aromatic plant in the field with rich in carbacrol, which is very active on ticks, aphids, and nematodes. And the Lavandula luceri endemic plant I mentioned before, we have domesticated in the field this plant, and finally we have a sustainable production of essential oils rich in necrodense active, as I mentioned before. For the optimization, for the fungal extras can be optimized by biotransformations and modified fermentations. Biotransformations require the enzymatic system of a fungi that can be applied to a major compound from a plant to obtain more active derivatives. Like in the case of the African neem, we obtained 2 derivatives which were very epoxides on the lateral chains, which were more active than the initial compound. Also, from endemic plants such as Bethencourtia palmensis, the fungal endophyte isolated from this plant has been fermented with different types of media and ingredients and additions to the media like glass wool, metallic mesh, or talcum powder for the production of neohydroxyaspergillic acid, which is antifungal, or melain, which is acaricidal. As you can see here, the different media gave different productions of the active compound, and also the addition of different enhancers in the media resulted in the optimization, for example, here with the metallic mesh of the production of melanin. For new applications, these biopesticides can be used for phytoimmunization. In our case, what we did is we took tomato seeds, we treated them with the essential oil of the wormwood that we have been cultivating in the field in a very, very low dose, and then we produced tomato seedlings from tomato seed coatings. These tomato seedlings were infected with Fusarium oxysporum, and what we obtained was that the control was okay because it was not infected. The plants treated with essential oil were similar to the control. The plants infected with Fusarium oxysporum were not developing, but the plants from seeds previously coated with the oil were resistant to the effects of the Fusarium after 12 days. When we looked at the metabolomic response of the tomato seedlings, we found that the terpene metabolism was modified in these plants, as well as the lipid metabolism was also modified with the seed coating with, as I mentioned before, a very, very low dose applied. So, like you can see, there's another approach. We are also developing new applications such as adapted formulations using carriers and also synergisms with essential oils, which are new applications. For example, we are developing the formulation with synergistic combinations to control the truffle, the beetle that eats the black truffle. Black truffle is a crop in Spain in places. It's a very specific crop, in the trees in the forest, but it's a crop. It's cultivated, and that has caused the emergency of a new pest, which is Leiodis cinnamomus. And there is no treatment available for the control of this pest, and everything that we use against this pest has to be natural because we cannot affect the truffle. Therefore, we have found different combinations of essential oils that do not affect the truffle, but they do repel the beetle. So we are trying to make the beetle not to colonize the truffle and develop damaged truffles at the end of the crop, which is very a lot of damage. All these results need to be transferred to the productive sector, such as companies. We are interacting with companies listed here, Aliovitro, Ecoflora, and so on. And we also protect the results. We not only make publications, but we also make patents to be able to transfer these results to the society and the productive sector so they can develop the biopesticides, which is our main goal in this research that we are carrying out. Finally, I want to acknowledge my collaborators from the University of Politécnica de Madrid with the biotechnology group, the aromatic plant domestication department in the Centro de Investigación en Tecnología Agraria of the Government of Aragon, and my group at the Instituto de Ciencias Agrarias, with the researchers, the students, and the technicians. And all of you for listening to my talk.
Thank you so much, Professor Colomna. So I would just like to say once again that we listened to Dr. Azuena González Colomna from the Institute of Agricultural Sciences in Spain, and she was speaking about pesticide problems and innovative solution in the context of the EU market. We would like to thank you once more for having this wonderful speech ready for us. And we will move on now to our next speaker, which is Professor Dr. Assunta Bertaccini from the University of Bologna in Italy. And her topic today is phytoplasmas still under studies plant pathogens. Dr. Bertaccini is actually a very distinguished professor of the plant pathology at the University of Bologna, Italy, and she started her career in 1997 when she led groundbreaking research on phytoplasmas and plant pathogenic bacteria. She is a member of many esteemed societies, including the Società Italia di Patologia Vegetale, American Phytopathological Society, International Organization for Mycoplasmology, and others. And she is also the recipient of the Emmy Killenberg Nobel Award, and she authored over 400 scientific publications. We would like to thank once more the Professor Bertaccini for joining us here for this talk, and we would like to invite her now to share her presentation. Professor Bertaccini, can you start sharing your presentation, please?
Thank you, and sorry for the inconvenience. Thank you for the opportunity. I will now move to a very specific field, a micro field, probably in plant pathology, but having quite a relevance. I imagine that the audience don't know a lot about this pathogen. I study bacteria, but very special bacteria that are phytoplasmas, discovered by Japanese group in 1967 in the phloem of the plant, showing a special symptom. They were not killed, they were not so damaged, but they show symptom like a witch's broom and a yellowing. These bacteria are transmitted by insect, and they can be maintained only. Sorry, there is a voice in the background.
Yes, I have muted everybody, so now it should be fine.
Thank you. Sorry for this. What I want to tell you is just a little about phytoplasma, just to make you aware of the problem. We are still not able to have this bacteria in a pure culture. So we have names now, but still as a candidatus, so provisional names. The phytoplasma are bacteria in the gram-positive group, together with a few other plant pathogenic bacteria like spiroplasma and clavibacter. They are pleomorphic because they lack the cell wall. They are very small. They can only be seen by electron microscopy. And their habitat is the hemolymph of insect and the phloem of the plant, for the reason they have no cell wall. They have a low ZC content, small chromosome size, and different kind of usage of the genome, and no sterol in the cellular membrane. Since 2004, we have the first whole genome of this bacteria. This is the Japanese group that was producing the first genome sequence. The one in the middle is me, but I was not involved. I am involved because I am leading a group worldwide working on this special bacteria. After this group sequencing, it was possible to have the first pathological confirmation about the role of this bacteria in the disease. As you can see here, you have on the left infected plant Arabidopsis by phytoplasma, onion yellow in this case. On the right, you have the specific gene Tengu discovered through the mining of the whole genome, producing exactly similar symptoms. So this is an indirect confirmation of pathogenicity that was followed by more sequencing of genome. And here you see another factor called SAP11 discovered by another group that is able not only to modify the shape of the plant, but is also able to modify the behavior of the insect vector, in this case, increasing the fitness and the fecundity in specific. So here you see some close-up of the phytoplasma under the electron microscope. The bottleneck for this studying this pathogen is their detection and identification in the plant, because to have a suitable management, we need to know all the components of the pathosystem, because it's not enough to clean up the plant from the pathogen. That is very difficult, but it is also necessary to avoid the insect vector that are carrying the pathogen from one plant to the other. So we cannot speak of real diagnostic for phytoplasma, but only of detection for the lack of fulfillment of Koch postulate. But in any case, there is a very good reason to believe that they are really associated with the disease we see around. You see here, for example, periwinkle, that is a test plant used for work on phytoplasma. And you see on the top left an healthy plant and how the infected plant look like. This is also an indirect method of detection of the phytoplasma in the plant by transferring to this plant by dodder or by insect. And after you see some insect vectors, some DAPI microscopy staining for the identification, not the identification, the finding of this bacteria in the phloem. But the jump on the study about phytoplasma was done by molecular biology that since more than 30 years now is allowing to differentiate them from each other without the need of cultivation. Here you see the final result of amplification and restriction fragment length polymorphism of the 16S rRNA gene that is now used in general for bacteria confirmation of identity. But we started for having the first differentiation of phytoplasmas in the 90s. As you can see from the profile, you have different profile, and different profile are related to different phytoplasma. In this case, are grape wine phytoplasma associated with the quarantine disease in Europe, that is flavescens doré. together with the 16S, other gene can be used for the differentiation of strain and the more detailed identification of phytoplasma. These are pictures taken showing the same series of samples digested with different enzyme and different gene, RP and SEC-Y. And you see that the group of profile, identical profile means identical gene, more or less identical, very similar. In any case, for the community working on phytoplasma, there is also the possibility to have a collection, access to the collection of phytoplasma and micropropagation that is maintained since 30 years by me in Bologna. These strains were first differentiated, as I showed you before, by RFLP analysis, and after they are classified with the Candidatus phytoplasma system that was published the first time almost 20 years ago, but recently we update the group working on this in the world, update the guideline for identification, because it is really important to know what is the phytoplasma infecting the crop in order to perform the management. In this moment, we have about 42 Candidatus phytoplasma species, and the rule is written here. I will not bother the audience with this thing that can be found easily in the literature. And we have identified Candidatus for a number of plant diseases. There is only one missing as a Candidatus, that is the flavescens doré phytoplasmas, because there are no sufficient differences from the molecular point of view from the already described Candidatus. But of course, the pathogen has differences for epidemiology and other features. Here you have the list of the available Candidatus published until now. On the left, you have the name of the disease, main disease from which they were studied. In the center, you have the name of Candidatus, and on the right hand, you have this number that are Roman number, that are the group, the ribosomal group and subgroup that were used initially, and in some cases are still used and useful to distinguish the phytoplasma from each other. This is the rest of the list, as you see, is a very, very long list. But this is not enough. This classification is based on only on the 16S gene identity. What is adopted in order to improve and to make it more precise is the use of other housekeeping gene. Listed here are only 5, but they can be more, because now we have a lot of genome available, so several genes are available for a number of phytoplasma. And by comparison of this gene, it can be possible also to differentiate the strain, and mainly the pathogenic strain. That is very important for studying. Some idea about how these diseases are in the world known. This is the first Candidatus phytoplasma named by the French group in the 90s. It's Candidatus phytoplasma aurantifolia, now renamed Citri, is associated with a disease called Lyme witch's broom that is seriously affecting citrus, especially in the Arabian Peninsula. In any case, it is not only affecting lime as the name, but also other citrus. Over the time, it was found also in other orange and other. Worldwide, one of the main diseases is lethal yellowing of coconut. This is an old picture taken from National Geographic, published in 1978, a picture taken in Florida. The disease is associated with Candidatus phytoplasma palmae, and it is killing the palm. I will come back to this a little bit later to show how large is this disease in the world. As I told you, the phytoplasma are insect-transmitted bacteria, and the insect that are transmitting belong to Fulgoromorpha, Cicadomorpha, and Sternorhyncha. Not all the insects are able to transmit, they should be flowing feeder. The main group transmitting the phytoplasma are Cicadelidae and Psyllidae. These are some of the phytoplasma vectors identified worldwide. As you can see from the list on your right, the same phytoplasma can be transmitted by more than one insect vector, unfortunately, to make our life more complicated. And there are several crops that are severely affected. You have the name of the candidatus phytoplasma on your left, and from the name you can more or less recognize the crop that is infected. A lot of fruit trees, grape vine, and also forest tree, and among the horticultural crops in the soil, the sugar beet is recently an emerging issue in Europe, at least. In the insect, the phytoplasma are acting as a real inhabitant. In fact, they also are transmitted through the egg. These are the phytoplasma combination in which the egg transmission was demonstrated. I will not go into detail. And phytoplasma are still more difficult pathogen because they are also transmitted through the seed. This is one of the fields that is less acknowledged worldwide, but the seed transmission was demonstrated in several crops listed here, like alfalfa, lime, tomato, cabbage, and corn, sesame, carrot, and the most recent is eggplant called brinjal. This problem is very heavy, for example, in India, where brinjal beetle leaf is the most important disease for eggplant. You can see in the center a picture of an eggplant with seeds inside that are already germinated. This is a clear sign of the presence of phytoplasma because they are jeopardizing the hormone balance of the plant, and the plant starts to germinate in appropriate period about seeds. And in the top center, you have an alpha-alpha symptomatic. Also for alpha-alpha is quite, I will show you now, for alpha-alpha, the seed transmission is very relevant. My group in Bologna discovered the seed transmission more than 20 years ago in seeds of alfalfa from the Sultanate of Oman. And here is what we did just last year. We still have seeds from the batches we tested in 2002, and we retested with a student. And what we found surprisingly is that one surprise was the seeds of alfalfa after 20 years still germinated. But the second surprise was in this seed, phytoplasma are still detected. I need to clarify that we test for phytoplasma not the seeds, but the seedlings. And we usually grow the seed in vitro to avoid any kind of possible insect transmission. So this is quite important because alfalfa is very relevant worldwide, but especially in the country where they are used for feed for animals. The presence of this problem is wide and helped the phytoplasma to be discovered and to be managed in some way in several crops. Just to show you something more, Candidatus phytoplasma aster is the same phytoplasma, but the symptom you can see associated with it can go from aster yellows, that is a green flower you see on the left, to Paulownia witch's broom, that is a tree. So the same phytoplasma is inducing different symptoms in different plant hosts. But on the opposite, this is cactus pear, Opuntia ficus indica. The symptom here is the excessive proliferation of the cladode, and worldwide, at least Italy, China, and Mexico, the same phytoplasma was found associated with this kind of problem. Of course, this plant can be nice to see, but they don't produce fruit. Other important disease, I will just mention, if somebody has question, you can ask after. Important in Europe is apple proliferation. You see from the picture, the apple become not only small, as you can see, but they are also very ugly to eat. The flower is not good because the phytoplasma are living on sugar. So they are using the sugar, and the sugar cannot be accumulated in the fruit. The other symptom are enlarged stipule, which is as the name of the disease, and the reddening of the leaves. Is present worldwide in Europe, and it is transmitted at least by 2 Cacopsilla picta and Melanoneura, and also by Fiberiella flori. As I promised, a little bit about the palm. Worldwide, wherever the palm are grown, there is a phytoplasma disease. In general, the disease is called coconut little yellowing because coconut is the most important among palm that is affected. You see the result on the top picture. The symptom is called the telephone pole because the plants drop the leaves and drop the fruit, and so they are really killing the economy based on this crop. But as you can see from the list below the name coconut little yellowing, different candidatus phytoplasma are causing this associated with this disease worldwide according to the different geographical area. For example, in the America, there is a candidatus phytoplasma palme, while in Africa is palmicola e cocos tanzaniae, and also in Australia, Papua New Guinea, is candidatus phytoplasma novi guineense, and also candidatus phytoplasma malaysianum in Malaysia. In the Indian subcontinent and China, the phytoplasma present are different again, is candidatus phytoplasma asteris. In any case, this is another nice symptomatology to be shown. This is a case of a little country in Africa that is Equatorial Guinea, where the poor production of coconut was completely destroyed by the disease. And this is a very recent case in the past years. And the work on this area is quite complicated. These are symptoms of coconut little yellowing in Florida, in Jamaica, where they have already 3 epidemics over the 30 years that is killing the plant. And in this area, it is also known that one of the insect vectors, at least the main, is Aplaxius crudus. In the other part of the world, it is not known the insect vector, but in Mexico, it was demonstrated that the seed transmission of phytoplasma. To another crop, we go to grape wine, and grape wine, I don't need to enter in many detail. You see the long list of country and phytoplasma detected. The grape wine is infected by phytoplasma everywhere in the world. Quickly, the 2 main diseases have a French name, Bois Noir and Travessens doré. They are 2 different phytoplasma, and they have 2 different kind of epidemiology. While the Travessens doré is only transmitted by specific grape wine feeding insect, that is Caphueria occitanus, the Bois Noir is transmitted by other polyphagous insects like Hyalestes obsoletus and others, and it is infecting not only grape wine, but several other crops. Here you see symptoms specific for both diseases. They cannot be distinguished. By the symptom, you have to go to molecular tool to distinguish which one, and in this case is very relevant because the management is quite different according to the problem of Bois Noir and Travessens doré. But I do not have the time to enter into detail. You see the killing of the plant on the top left, the lack of lignification, the bunch without correct ripening, and the killing of the little berries, and also the change in color and shape of the leaves. But phytoplasma usually are a problem because, especially now that we know how to detect them, it was possible to see that they are present very often in mixed infection with other bacteria. I mentioned at the beginning the sugar beet, and in this moment, in the majority of the sugar beet cultivation in Europe, there is a large and extensive problem that is linked, associated with the presence of bacteria, and in particular of phytoplasmas and other bacteria. But it's not only a problem of sugar beet. You see here some picture of sugar beet, potato, and citrus, and several important diseases like one long bean in citrus, zebra chips in potato, and Bacillus shells in sugar beet are associated with the presence of phytoplasma and other bacteria, still candidatus, but different gram-negative, Candidatus liberibacter, Asiaticus africanus and americanus for citrus, Solanacearum for potato, that is a big problem in South America now, and for sugar beet, together with Candidatus arsenophilus phytopathogenicus, at least in some part of Europe, together with Candidatus phytoplasma solani and asteris that are detected together. So the problem is really to be able to detect the pathogen and to act in some way against them, to have some kind of instrument to cope with this pathogen. It is important to have them in pure culture. I told you that this is not yet available, but my group worked for, let's say, ever about this task. And we get some kind of result published already 10 years ago about the obtainance of colonies enriched in phytoplasma on artificial media. Why I said enriched? Because they are not only phytoplasma in some cases, there are also other bacteria that are in general endophyte. Here you see colonies on the top, and we have obtained these colonies from coconut or from citrus or from grape wine directly infected in the field. It was the best way we have to get these colonies and to have them at least for a number of months in cultivation and to use. In this case, for example, we have these colonies obtained from citrus. This was a project funded by European Union called Tropic Safe. You can find the result still in the web until 2032. So if you are curious, you can look at this. These are colonies obtained from citrus showing one long bean symptom in Cuba. What is interesting, besides to have both pathogen, Candidatus liberibacter and phytoplasma, is that in these colonies, we also detected colonies of phytoplasma of lethal yellowing. In reality, in Cuba, there was a big epidemic of lethal yellowing who killed all the seed of the palm. But in some area, we were able to find that the pathogen moved from the palm to the citrus. So this is quite an important information. This is the story of the endosymbiont and the endophyte that are growing in colonies together with phytoplasmas. And the last in the list of this finding is the one you see like a nice fried egg. In reality, is a colony containing phytoplasma and pseudomonas. This was isolated by one of my last PhD students from Ziziphus jujube, witch's broom infected in vitro. And the data of metagenomics showed that all the shoot, healthy and infected of jujube, were having these pseudomonas as endophyte. And what we suspect is that the pseudomonas was living and helping the phytoplasma surviving in our culture. Here is the metagenomic data. You see the relative abundance in citrus, for example. We have both Candidatus liberibacter and phytoplasma, but we already saw it in cultivation, so the tool is useful. So all this information are important to help the management of this disease. Management is based, as for the virus disease, mainly on prevention. Old method, but still effective. So the use of phytoplasma-free material when you produce your orchard. Elimination of symptomatic plant is not sufficient. I will show you later. Identification and elimination of insect vector when you know the insect vector, and also elimination of alternative host plant when you find the species of alternative host plant. Not possible to do this kind of generalized spraying with pesticide is against every law, at least in Europe, and every 1F practice. What we are developing since several years in Bologna with colleagues from engineering department is the use of plasma activated water. I will not enter into detail, I can explain, but there are published data about this. What I want to show you is what happens when we treat with this. It's quite a laborious technique, but you can go from a symptomatic plant to the asymptomatic plant. And this is what happened to at least in one case. You see that the plant in year 1 was dead, is a grape wine dead. After 1 year of application of this water that is injected into the plant, not sprayed for the moment. And you see that in the year 2, the plant started to sprout again, and we were also able to find a little bunch. So this kind of treatment is not a pesticide because it's water. And there is no phytotoxicity involved. So we are now extensively trying in the field to see if this result is consistent and reliable.
So sorry, Professor Bertaccini, I'm just going to give you a heads up that we are almost 3 minutes past your time for presentation. So
I'm sorry.
I would just like, it's okay. I would just like to kindly ask you to maybe come to the conclusion pretty soon.
I am.
Thank you so much.
This is the last slide. I'm sorry.
Okay, great. Thank you so much.
I just want to show you these are the same plant, half infected and half healthy. The healthy looking is still infected, so it's carrying phytoplasma. So this is the message. The plant are, even if asymptomatic part, don't prune, is not enough. The asymptomatic part can carry the disease, and so there's a lot of work to do. This is just the list of my collaborators through the years, since you see how many, and I'd like to invite everybody maybe interested to join in 2027 to the meeting we will have in Italy about this topic, that is the phytoplasmologist working group meeting. You have the web page where you can find more information. You see it's the 6th meeting we are having every several years in order to have this alarm in the country. Thank you, and sorry for taking more time.
Thank you, Professor Bertaccini. I completely understand. I would just like to thank you once more for taking time of your busy schedule to give us this presentation today. So this was Professor Dr. Assunta Bertaccini from the University of Bologna in Italy, and her topic was on phytoplasmasis, still understudied plant pathogens. Our next speaker for today, last but not least, Professor Dr. Hikmet Budak from the Arizona Western College in United States. So Professor Hikmet Budak is a director and professor at the Arizona Western College in Yuma, and he has 25 years of the experience and has accumulated extensive experience in academia and industry worldwide by working across various disciplines. His primary focus in his career has been on developing, understanding, and applying multi-omics and CRISPR editing tools for the crop breeding and genetics. And his topic for today is application of artificial intelligence to microomics. So Professor Budak, are you ready to give your presentation?
I am. Good morning, everyone, or good afternoon, or good night, wherever you are. It's 4:25 here, but in the United States. Anyways, thanks a lot for the opportunity. I would like to just stick with the time, and then I'll try to explain what I like to share with you guys. So today I'm going to talk about just application of the AI and ML to microorganism or microelements, non-coding RNAs, in response to some diseases and insects. But I have always a mission that wherever I go and then present, and I said the good life needs really good plants. This is important. So I'll start with one of the quote. I love that by Alex Haley. Anytime you see a turtle up on the top of a fence post, you know that he had some help. But I like to acknowledge all my team, and really I got a lot of help from my students, my teams, and staff and faculty members. And I like to acknowledge all of our funding agency for generous support as well. Without their support, I couldn't accomplish, we couldn't accomplish some of the research that we conducted. Currently, that our team is this, and then basically I have 2 full-time faculty members in addition to 10 adjunct faculty members and some technician and lab foreman. I also just established an international advisory team and from Australia, France, and Canada too. And I will start with some of the projects currently that we are working on, developing a workforce and trying to develop and professionally grow the next generation leaders and really plant scientists and plant breeders and plant geneticists. And basically what we are doing is that with that grant, and we got it from USDA, is just matching the mentees and mentors, and so that they can learn from each other, but also that they can start hiring and recruiting some of the students that we have in the industry. I'll briefly talk about some of the greenhouse and farms that we are currently available that we can easily use and utilize. And also we are open to some of the small research for public, which we can just help them, and in addition to some private companies as well. I'll start with the drought. Drought is a lifestyle disease, and if you look at it, and everybody knows that in 2016 or 2006, World Economic Forum just said the water crisis is one of the top important issues right now in the whole wide world, in addition to diseases and insects. So we will talk about the insect and diseases, but I just wanted to mention that the drought is really getting big lifestyle diseases as well. If you were to look at the top right corner of the picture, that we are expecting 9.6 billion in 2050. This is a fast slides that we see. What does that 9.6 billion people mean? I ask everywhere, and actually that don't let the numbers fool you, because we are expecting 12 billion people because they will eat like 12 billion people due to increasing income and the population as well. So we need to target that in 12 billion people instead of 9.6 billion people. Water, for instance, I don't know if you can see it. I can just show you that the little tiny water, that whole wide world that we have currently. And also that we can give the numbers, a lot of statistics that in disease responsive loss and insect responsive loss. And I've been working in wheat stem cell life for about 6 years. And one of the most important part in Montana, in 3 different states actually, Nebraska and Colorado, and we are losing 80 billion or $80 million every year. So you can look at that. What is the industry? This is the wheat stem sophili insects, and this is the insect on the left side, and then this is the larvae that inside the wheat. That is what we were dealing with. And currently that in Montana, some of my team in Montana BioAg, which is one of the private companies, we try to just develop some kind of biopesticides, especially against the wheat stem sophili and flea beets and canola. And also we have some biofungicides, especially for Syrian head blight in wheat, rust in wheat, again, yellow rust, and also the canola and pulses. There are some important diseases that we are trying to work on as well. We have some biostimulants that we are focusing on as well, mainly the fungus and for drought and heat tolerance too. I was going to put that slide at the very end, but I want you to just show you that what my mission and vision and what we need to do for the future, not only for research wise, but also that the developing and helping and professional growth of our students and next generation leaders. Actually, we need to develop some kind of curriculum and research that is encompassing that whole multidisciplinary, interdisciplinary, even transdisciplinary work and projects in order to translate to data into knowledge. And you can look at it, and mainly that the red boxes that you can see, that the network analysis modeling is one of the things, and experimental design is another one. Although we do have a lot of gene expression profiling, metabolome, and genotyping available, lots of precision ag tools is available today that we are in the college as well, that with the University of Arizona and Arizona Western College, we are developing and implementing some AI to use the drones. And it's easy now, available everywhere, and then they can easily use it. This is the curriculum development in Arizona that we are working collaboratively. and have a collaboration with the University of Arizona, some other colleges as well, to build some kind of curriculum that is going to be really helpful for implementing AI, understanding the agricultural system and diseases in insects, how we can utilize this. And then one of the important item that I just said is that we need to work together, that the translating in data into new knowledge for the new generation, our students. Today, I want to give you some update in our research. We developed some kind of programs, starting with the microRNA annotation, the long non-coding RNA annotation, and finally that we finished the small RNA annotation with some of my groups members. And this is now easily that you can do some of the analysis using artificial intelligence and machine learning using your laptop. It was really hard. And if you remember that the sequencing of human genome, it took 10 years with the 60 different lab to finalize and draft sequences. Today, it's just easy, just a couple of months is going to be. So let's look at the functional analysis and elements. In addition to some genes, I was talking about the long non-coding RNAs. These are some parts that you can see on the left part, and microRNAs. And there are more RNAs or small functional elements that is important for identifying the genes or regulating the genes as well. Genes is not just the regulating by itself. We know that in the past that we were talking about the Tata boxes and promoters and everything. There are a number of other functional elements. But what we identified and learned now is that long non-coding RNA or small RNA are also regulating the genes, either silencing or knocking down or just overexpressing the genes. So what we did is that we started with this idea, and then some of my students took over and then just get generated data. Our idea is just the presenting that, developing some kind of databases and for small RNAs, and that database is going to have some kind of future elements that is going to be separated each other and see that what are the targets. And after that, this is just the basically machine learning that I learned, and then I wanted to apply the small RNAs and with some of my students and postdocs and faculty members. And what we identified is that we can easily enter that the new data, new small RNAs, annotate at the end of the data as well. Two most important things that is important, since they are really small, that we wanted to understand that if we have a false negatives or true negatives that we wanted to identify, and what are the true elements that we need to identify. If you look at the rock curve on the right side, what we are trying to do is that we are trying to look at the perfect true positive rate, which is on the top one, and then this is what our goal is. And we compared all of our all commercially available tools with our tools to see that if it is working perfectly fine or if we have more false positive rates as well. So all we did is that we used and identified some of the algorithm that are available currently and compare with ours as well. What we identified is that with one of my students, currently she's in Stanford University as a bioinformatics scientist and working on that different things right now, is that what she identified, what she generated in the data is that our algorithm, which is like a long non-coding RNA LNC machine or MiR machine as well, microRNAs, it was much better than the available currently data or algorithm as well. So we looked at the machine learning algorithm using the different wheat data set, and which is like developed by International Wheat Genome Sequencing Consortium in 2018 and also 2019 for the tetraploid wheat. And we were all involved with these two sequencing data efforts. What we identified is that you can look at the hexaploid wheat and tetraploid wheat. Our LNC machine, also MiR machine as well, that was really higher compared to any other data or algorithm available, and we just identified the less false positive in our data sets using the algorithm that we identified or we developed. hopefully it is going to be commercially available. We are developing in some kind of like software so that everybody can use it. So we applied all these AI machine learning tools to whatever that we have in terms of insect responsive or disease responsive or drought responsive microRNAs to see that if we can eliminate the false positive and then focus on true elements. And one of them that I want to show you that in the beginning that we identified some insect responsive long non-coding RNA. You can see that we found a different numbers or percentage in that weed varieties that we used after sequencing. And basically that different varieties show that different number of microRNAs, and also those are not microRNAs or mimicking the microRNAs. That's what we identified using the machine learning system. And we all mapped the newly identified microRNAs in the different crop species too, wherever that they are located. And this is one of the insect responsive or solid stem varieties, microRNAs, and where it is located and which variety is located as well, compared to rice, rye, barley, different crop species, so that we can identify that this microRNA is precisely targeting the genes or targets that we are interested in. For instance, Fusarium head blight, or wheat stem sulfuri, or yellow rust. And currently that we are working on some lettuce, which is that Arizona gave me a lot of information about the lettuce Fusarium that I'm still learning about some green leafy veggies here in Arizona as well. And we identified a number of genes or microRNAs in different chromosomes and compared with rice, wheat, and barley. So this is a comparative analysis in different crop species so that we can identify common microRNAs commonly targeting that or precisely targeting the same target genes in different crop species. And if you remember that the first slide or second slide, I was talking about that what we need to do is just one is that's modeling or network analysis. If you are looking at that, the different network analysis, which microRNA is targeting the different microRNAs or long non-coding RNAs, and it is important to see the whole picture. This is a little bit complicated or busy slide, but I'll show you this. Let's say that one of the microRNAs is in the beginning, in the center of that whole small RNAs. You can see that the different colors, this is clearly telling us that what microRNA is networking or helping the other long non-coding RNA or mRNA, so that we can identify if we are deleting or knocking down or editing using the CRISPRs or any other editing tools. So how many genes that we are editing, or just if we are targeting precisely what we wanted to target. As you know that the CRISPR and also some other editing tools is just off target is one of the biggest issue. So looking at the network analysis, we can identify that when we edit one of the small RNAs, what we are also editing that the long non-coding RNAs or mRNAs, so that we are easily identify or decide if we can just clearly knock out some whole network or just a single microRNAs as well. This is the lettuce that I was going to show you that some of the Fusarium is important. There are 4 different Fusarium fungi, maybe that there's more right now and easily that people are really identifying. And Fusarium head blight in cereals is different than the lettuce. And this is different fungi, but we wanted to identify that or look at what our fungi found in cereals is also affecting or helping or the lettuce that to get rid of the Fusarium in lettuce too. We're currently working on that so that we can just identify there's some biological control, but also we identified there's some fungi that is really helping in cereals as well. So our recent study with my group, again, that is some of my students and colleagues, that what we did is that we worked on that for 2 different main wheat varieties. We identified some microRNAs, and we also did some run the RNA, mRNA analysis or transcriptome analysis as well. So we looked at what are the microRNAs that are really upregulating or downregulating, or number of microRNAs is upregulated because of the Fusarium head blight, or downregulated because of the Fusarium head blight. You can see that the WIDA, which is one of the varieties, it's controlled, which inoculated, and it's not disease infected. But you can see that the WIDA, when you infect with the Fusarium diseases, number of microRNAs is really increasing. And Hank is the same way, but the number is different between Hank and WIDA. And I can talk about each one of the numbers and then names of some of the microRNAs, but the important part here is that finding the number of microRNAs, but what microRNAs is really responsive in response to Fusarium. And Fusarium head blight and Fusarium gramineum is one of the ones that we also worked on this. So basically, these are the microRNAs in WIDA control and infected, and Hank control and infected ones that are really expressed. So we identified some common ones. Now we are currently editing using the CRISPR. And some of my team right now is Montana Bioag. We haven't started here right now for the CRISPR editing, basically that we wanted to identify, or we wanted to edit some microRNAs that are directly targeting that Fusarium head blight in wheat stem cell or in wheat. And actually, some of the Fusarium responsive microRNAs is valid for wheat, but also that barley, oat, and rice, and rye, and durum wheat too. So basically, that we identified the 10 main common microRNAs currently that we are trying to edit and knocking down the genes if it is possible. And if we identify the one negative allele, that is going to be hopefully that we're going to get rid of that gene. And eventually that we will have some kind of new varieties, and that is going to be helpful for the farmers and growers and industry group that is free from the Fusarium head blight. And also that my goal and my vision here is as well that we would like to identify some of the genes that is affecting the Fusarium diseases in lettuce so that we can eliminate and we can move forward. With that, I think I'm right on time. Thank you so much. And this is one of the slides that I adapted from my student. And I'd like to thank you so much. And I would be more than happy to answer any question that you might have.
I would like to thank Professor Budak for holding this presentation and for accepting to be our speaker today. So once more, this was Professor Dr. Hickman Budak from the Arizona Western College of United States, and his topic was an application of artificial intelligence to MiRNomics. We have actually come to the end of our presentations for today. So I would like to ask kindly speakers to ask their questions in the Q&A box. We will give you a couple of minutes and you can ask all of the speakers for today whatever you wanted to ask and while the presentation was held. I would also just like to emphasize that it would be very helpful to address the professor that you wanted to ask the question to. So please feel free to extend the questions in the Q&A box. Thank you so much. We will wait for a minute or two for everybody to write their questions. As we wait for the attendees to write a couple of questions in the Q&A box, if professors have any questions for their colleagues, feel free to ask.
Just want to say thank you so much. I learned a lot today, really, from all the presenters, and thank you so much. That was great, and it was a great learning part for me.
Yeah,
Thank
it
you
was
so much.
great for me too.
It was very nice. I will have a question for Professor Assunta. Do you know if there is some differences between the Huanbin long that is in Florida to the strain that is here in South America?
Oh, as far as I know, I should go to Brazil, but I just delayed my travel. In any case, the one long bean in Brazil is having two different candidatus Beribacter. There is also one specific called Americanum that was selected, let's say, or sprouted near São Paulo. So this is one main issue. And also in Brazil, phytoplasma were detected, while in Florida, as far as we know, in Cuba, yes, I worked in Cuba, but I don't know in Florida. I think only the other is present. I don't think it is critical because I suspect that diaphorina is transmitting all of them, but is not something is under my knowledge. In any case, I thank all of you because also it was interesting for me.
I would just like to thank everybody for taking time of their busy schedules to become a speaker today. And thank you so much for attending the MDPI International Day of the Plant Health 2025 to all of our attendees. I would like to note to the attendees that they can also send us their questions later on. Once the webinar has ended, you can send us via email and we will then transfer it to our invited speakers. So thank you everybody for joining us.
I have one more question, Sorana.
Of
Will
course,
be
Professor
the
Heisen.
recording of the webinar available for openly?
Yes, the recording will be available in a couple of days. We will try to do this as soon as possible, and you will be able to see it and download it from the webinar website. We will also send you this in an email.
Okay.
Thank you.
Okay.
Very
Thank
welcome.
you.
So as
And
Professor
thank
Budek
you
has...
all the colleagues for the very interesting talks they deliver. Thank you very much.
Thank you.
So thank you so much, Professor Heinzen. I would like to thank everybody once again for joining us here and to remind you to check the website of this webinar to download your certificates. Once you log in, you will be able to do that manually and also to check the video. And everybody have a good day, good night, and a wonderful morning. Goodbye.
Thank you so much. Bye-bye.
Bye.